I welcome you to this presentation on the laboratory handling and processing of leaf samples for disease diagnosis. We are actually aiming at ensuring accuracy and reliability in our tests. First, you need to collect samples and then you need to prepare them. In collecting samples, you need to select symptomatic leaves representing various stages. You also need to avoid damaged or contaminated leaves. Of course, you have to label them appropriately based on date, plant species, and location. And you also need to remove soil and plant debris and cut them into manageable species while preserving the portions that are exhibiting symptoms. Of course, you need to also clean and label the containers containing the samples. In terms of sanitization and storage, you need to disinfect the tools and surfaces where you'll be working and be changing gloves in between to avoid cross-contamination. You need to also store and transport the samples, keeping them cool and moist. And if processing will be delayed, you need to refrigerate them. And in processing and interpretation, priorities must be given to samples of urgency. And of course, you have to follow the standard protocols for processing. You have to consider the plant history, the environment, and the plant species and the type of symptoms, even in your diagnosis. And then, of course, there may be need for you to consult diagnostic manuals or experts for accurate interpretation. And having done all, you need to communicate your results promptly and clearly, provide recommendations for disease management, and of course, in conclusion, you need to follow proper handling, processing procedures, and ensure accurate disease diagnosis and effective disease management.